press the bell icon on the YouTube app and never miss another update.
Hello everyone. So this is my last video on the chapter Cell, the Unit of Life. So in this video I am going to discuss certain multiple choice questions which may help you to tackle all the competitive exams like the KCET and the NEET. So to start with, the name of the chapter is the Cell, the Unit of Life. So here in this chapter we have studied all the cell organelles. So the difference between the prokaryotic cell and the eukaryotic cell, and the various organelles and their functions present in both prokaryotic and eukaryotic cells. So to start with, take the question, the first question to be discussed: in which one of the following organelle are non-genetic RNA molecules synthesized? So the question is, in which of the following organelle? So we know what are organelles. The various parts present inside the cell are called as organelles. In which one of the following organelle or non-genetic RNA? So I hope everybody know what is RNA. RNA is a nucleic acid. DNA and RNA are two important nucleic acids. And whenever DNA is, almost all living organisms, either DNA is present or RNA is present. If DNA is present, DNA itself is the genetic material, where the genetic material, in the sense they are the units, hereditary units, which are carried from one generation to another generation, that is from parents to offsprings. So now the question is, when DNA is present, RNA has no role in hereditary, as a hereditary unit. It has certain other functions in the body. Whereas if DNA is not present, RNA itself will act as the genetic material or the transmitting units from one generation to another generation. And now when it is there is a word like non-genetic RNA, that tells that DNA is almost present in the cell. And when DNA is present, DNA is the genetic material and DNA is capable of producing the RNA. And we all know, we have studied eukaryotic cells. So we have studied what are the functions of nucleus. So in the nucleus the DNA is present. So DNA will produce RNA, means that nucleus is the right answer. So the organelle which is involved in the synthesis of non-genetic RNA means that is nucleus. So non-genetic RNA, here we have three types of RNA, the mRNA, the tRNA and rRNA. rRNA is ribosomal RNA, tRNA is transfer RNA and mRNA is the messenger RNA. So all of them, they are synthesized in the nucleus by the DNA because it has got various enzymes to synthesize them. Okay, next question. In which of the following organelle, in 70S and 80S ribosomes, S stands for? So here 70S, 80S. 70S is once again made up of 50S and 30S, two subunits, and 80 is made up of 50 and 40. 60 and 40. So S here stands as the sedimentation coefficient, which is called as the Swedberg unit. S stands for sedimentation coefficient, which is called as the Swedberg unit. It is not an SI unit. So sedimentation means, see, these are particles. So the heavy particles, they settle at the bottom. So during the process of after centrifugation, based on the rate of centrifugation, they get sedimented at the bottom of the container. Based on this, it has been told that sedimentation coefficient, and therefore S here stands as the sedimentation coefficient, which is also called as referred as the Swedberg unit, a measure of the density, you can tell. Next question. So answer 1 is the right answer, which tells S is sedimentation coefficient and Swedberg unit. Next question. In most fungi, the cell wall is chiefly made up of. So cell wall, which is present only in the plant cells and all the other kingdoms, the Monera, Protista, the fungi and the plantae and the animal kingdom. So animals do not have a cell wall, as you know. The outer membrane, most is the plasma membrane or the animal uh, plasma membrane. So here the cell wall is made up of a substance called as chitin. Cellulose is the cell wall is made up of cellulose in case of plant cells, and chitin, it is in case of fungi, the cell wall is made up of chitin. Protein and lipid, they are components of the plasma membrane rather than the cell wall. So the cell membrane is made up of lipoprotein, we call it as. So lipoprotein is nothing but lipid and protein. So the right answer is chitin, option 2. Next question. Which one is not properly paired? Question number 8. Which one is not properly paired? Look at the first option, the Golgi apparatus, breaking of complex macromolecules. Second is rough endoplasmic reticulum, which is involved in protein synthesis. Mitochondria, oxidative phosphorylation. Smooth endoplasmic reticulum, lipid synthesis. So as we already know, the functions of the organelles, Golgi apparatus is rather called as the packaging material. So it is not involved in breaking up of complex macromolecules. The breaking up of complex macromolecules is brought about by the hydrolytic enzymes, and that function is performed by lysosomes and not Golgi apparatus. So the not properly paired is one, that is Golgi apparatus breaking of complex macromolecules is a wrongly paired or mismatch. I should tell that mismatch. It is a mismatch. The second one is rough endoplasmic reticulum, which is involved in protein synthesis, is of course right, because rough endoplasmic reticulum, also called as RER, because they are called RER because they are studded with ribosomes, and ribosomes are the sites of protein synthesis, as you all know, and it is of course involved in protein synthesis, so it is rightly paired. And the third is mitochondria, oxidative phosphorylation. So we know that mitochondria is the energy producing cell, or the powerhouse of the cell, and it produces energy and stores energy in the form of ATP molecules, that is adenosine triphosphate, and this is produced by one of the steps is oxidative phosphorylation, which takes place in the mitochondrial matrix. So it is also a correctly matched pair. And the fourth, smooth endoplasmic reticulum and lipid synthesis. So that is also the right function of SER. Therefore, the right answer is option 1, Golgi apparatus breaking of complex macromolecules is a mismatch. Next question. Question number 10. Question number 10. Most abundant lipid in cell membrane is. So the various lipids they have given over there, sphingolipids, glycolipids, phospholipids and cholesterol. So now we have already studied the Singer and Nicholson model of cell membrane, the fluid mosaic model of the cell membrane we have already studied, and it comprises of the various lipids. And we have, in the diagram itself, we have written glycolipids, phospholipids, and cholesterol is also a type of lipid. So the one which is absent there is the sphingolipid, and the most abundant is the phospholipid, which is present in the cell membrane is the phospholipid. The lipid along with phosphorus, you call it as phospholipid. Next question. So the answer is option 3. Next question. Of the following types of organism which do not have a membrane surrounding their chromosome. So chromosomes are present in the nucleus, and the nucleus is not bound by a membrane. Look at the question. Of the following types of organisms which do not have a membrane surrounding, instead of asking nucleus, they have asked chromosome, and chromosome is present in the nucleus. So if they are telling they are not, the chromosome is not bound by a membrane, means it is, the meaning is the nucleus is not bound by a membrane. That means it is prokaryotic cell. So now let us read the question once again. Of the following types of organism, you have to identify which are the prokaryotic organisms, right? Because The one which do not have membrane surrounding their chromosomes are called as prokaryotes. So you identify which are prokaryotes. So archaebacteria, the options are archaebacteria, eubacteria, fungi, protozoa, blue-green algae. Archaebacteria means they are ancient bacteria. Eubacteria means new, newly formed bacteria. Fungi we already know which comes under the kingdom Mycota. The bread molds like rhizopus, yeast, penicillin, all these are with the various fungi. And protozoans we already know that they are the unicellular organisms like amoeba, plasmodium, etc. And now the question, you have to identify the prokaryotes. Of course, archaebacteria or the one which are called as methanogens. You might have studied in the second chapter biological classification about methanogens, acidophils, halophiles. All these are archaebacteria which are prokaryotic. So archaebacteria are prokaryotic and many examples coming under eubacteria, that is some bacteria, some types of bacteria are also prokaryotic, and the blue-green algae, the BGA, all of them are prokaryotic. So the option is fourth option, A, B, E, that is archaebacteria, eubacteria and blue-green algae, or the organisms which do not have membrane surrounding their chromosome is the right answer. So next question. The vacuole is lined by a membrane called. So it's a direct question. So the vacuole, it has got a bigger lumen which consists of the various waste materials which are not required for the cell, and this vacuole is lined by a membrane which is called as the tonoplast. So to confuse you, they have given two important terms there, tonoplasm and tonoplast. Don't get confused. Be confident it is tonoplast. Next question. Question number 14. Cell is considered as a self-contained biological unit because it is bound by plasma membrane, contains all living organelles, is capable of independent existence and self-perpetuation, has all above mentioned attributes. So cell is a self-contained biological unit. So we know it is the basic structural and functional unit. Now the question why cell is considered as a basic structural and functional unit comes in front of us. Is it because it is bound by a plasma membrane? Let us look for a better answer. The second option tells because it contains all living organelles. Maybe. Let us look into the third option, is capable by independent existence and self-perpetuation. This holds most better, so we can click, you can pick the third is the right answer. Has all above mentioned attributes. So this is most appropriate. It is capable of independent existence and self-perpetuation, because you see all unicellular organisms have all these characters. They are capable of independent existence and they are capable of reproducing also. Therefore, a cell can be considered as a self-contained biological unit. So option 3 is the right answer. Next question. Based on, ribosomes or granules made of. So ribosomes they are present both in prokaryotic and eukaryotic cells, and these ribosomes are of different types, as we have studied 70S and 80S are the major two different types, and these ribosomes are also present on the endoplasmic reticulum in case of eukaryotes. So ribosomes, they are granules which are made up of, so rRNA and proteins is the right answer. rRNA is the ribosomal RNA and proteins. So the right answer is option 3. Next question. Based on type of pigments, plastids can be classified into. So we have studied about the plastids which are present only in the plant cell and not present in the animal cell, and these plastids are involved in photosynthesis, and there are 3 types of plastids: the chloroplast, the chromoplast and leucoplast. So option 3 is the right answer. But still, let us try to look into the other options also. The first one tells phytochromes. So phytochromes are nothing but the various pigments present in the plants. Phycobilins and phycoerythrin are two important words there. Phycobilins are also various pigments present in the plant cells, and phycobilins are of two types. One is phycoerythrin and the other is phycocyanin. Phycoerythrin contributes for the red color and phycocyanin contributes for blue color in various plants without green leaves. The second is amyloplast, elaioplast and aleuroplast, or three types of leucoplasts which store carbohydrates, that is amyloplast. The elaioplast, they store oils in them, and the aleuroplast, they store proteins in them. Next, the option 4, chlorophylls, carotenoids and xanthophylls. So as we know, chlorophylls, they are green colored pigments. Carotenoids, they almost contribute for orange color, and xanthophylls, they contribute for yellow color. So all are various coloring pigments present in the plant cells. So, but the question, for the question given over there, the right answer is option 3, chloroplast, chromoplast and leucoplasts. Next question. Microtubules are made of. So microtubules, they are the proteinaceous filament-like structures which are present embedded in the cytoplasm. So they act as the cytoskeleton, and these microtubules are made up of the options given over there are actin, keratin, tubulin and dynein. So actin, they are present in the muscle cells, they are proteins, and keratin, they are present in the skin, and tubulin is the right answer, where microtubules are made up of proteinaceous units called as tubulin. So option 3 is the right answer. Next question. Which is wrong? Both chloroplast and mitochondrion have an internal compartment or thylakoid space bounded by thylakoid membrane. Then both contain DNA. Chloroplast is generally larger. Both are covered by double membrane. So the question is, which is wrong? So both chloroplast and mitochondria have an internal compartment or thylakoid space bounded by thylakoid membrane. Only chloroplast have thylakoid space and thylakoid membrane, but in mitochondria there is cristae, and on the cristae there are F1 and F0 particles. So elementary particles are present in the mitochondrion, and this thylakoid space and thylakoid membrane is restricted only to the chloroplast. Therefore, that is a wrong statement. But still, let us analyze the other options. The option 2 tells both contain DNA. Of course, both mitochondrial matrix and in the chloroplast there are DNA molecules. When the third, chloroplast is generally larger, of course, chloroplast or convex like structures which are larger when compared to mitochondrion, the rod like structures, and both are covered by double membrane. Of course, both have double membrane, the outer membrane and an inner membrane, and between them there is a space also. So all the second, third and fourth are right statements. The first statement is wrong. Therefore, the right answer is 1. Next question. A hook, basal body and shaft are related to. Long tubular hair-like structure on bacterial surface are called. Non-membranous phosphate granules glycogen in bacteria are known as. Enfoldings of plasma membrane in bacteria are. Complete the above sentences with the following words. So you have to tell what it is. They have given certain characteristics of the organelles and you have to tell what it is. So question 20, the option A, the statement A tells hook, basal body and shaft are related to. They are all related to the. Is there option? Option on the other sheet. Only 2 options are there. I will go back to the question part. So there A and B, I don't think it will match. A hook, basal body and shaft are related to, of course, they are related to flagellum in both option 1 and 2, which we can see over there. They have given pili. We have to look into the third and fourth option. So next is long tubular hair-like structure on bacterial surface or called, you can put it as pili, and infoldings of plasma membrane in bacteria are nothing but mesosomes, and the non-membranous phosphate granules, glycogen in bacteria are nothing but the inclusion body. So according to me, A is flagella, B is pili, 3 is inclusion body, and fourth is mesosome. Let us look into the third and fourth option. So A flagella, B pili, C inclusion bodies, D mesosomes. Option 3 is the right answer for this question. Next question. These structures are necessary to produce spindle fibers during cell division. So I have also started with the chapter called as cell division. These structures are necessary to produce spindle fibers. So while learning some of the functions, we have referred to the word spindle fibers. So with that pretext, I would like to quote the answer as microtubules. So microtubules, they act as the spindle fibers during cell division. It is not the Golgi complex. It is not the centrosome. Centrosome, it will divide into two cylindrical structures. So centrioles and microtubules form the spindle fibers, whereas centrioles, they form the asters, and ribosomes, they are involved in protein synthesis. So the right answer is microtubules, which forms the spindle fibers during cell division. So the chromosomes, they just stick to the spindle fibers during the cell division, and all those aspects we'll study in the chapter cell cycle and cell division. Next, but here the microtubules, one of the function of microtubules is it produces the spindle fibers. Next, arrangement of microtubules in centriole is. So in the centriole, the microtubules are arranged. So in the peripheral region of the centriole, so centriole has got a cart and wheel like structure, it has got the centriole, which are present only in the animal cells and absent in case of plant cells. So in the peripheral region, there are 9 microtubules with 3 tubulin structures with 3 tubulin protein structures. In the center there is a central hub, and the central hub is connected to the peripheral microtubules with the help of radial spokes. So this type of arrangement, you call it as the cartwheel arrangement in centriole, and that shows 9 plus 0 arrangement. So 9 plus 2 arrangement we have studied in flagella or cilia, but 9 plus 0, that is in the center there is a hub, there are no microtubules, but only in the peripheral region you find the 9 microtubules. Therefore the right answer is option 4. Next, a cell organelle X is divided into 2 types on the basis of a cell organelle Y that helps in the protein synthesis. Identify X and Y respectively. Look at the question. A cell organelle, they are not naming it, they are just telling it is cell organelle. It is divided into 2 types on the basis of cell organelle. So cell organelle itself is of 2 types, they are telling. So we can directly suspect it might be endoplasmic reticulum because it is of 2 types based on the presence and absence of ribosomes. They are rough endoplasmic and smooth endoplasmic reticulum, that is RER and SER. So let us take S as endoplasmic reticulum, and let us suspect what is Y. What is Y? Y that helps in protein synthesis. So let us think that X is endoplasmic reticulum, and Y that helps in protein synthesis means it is ribosomes, because ribosomes are the sites of protein synthesis. So the answer is endoplasmic reticulum, that is ER and ribosome. So option 3 is the right answer. Next question. Which of the following groups have a cell wall? So organisms with a cell wall and organisms without a cell wall. Look at the first option, bacteria, plant and animals. Of course, animals do not have a cell wall, so we will cancel that. And the second option, bacteria, fungi and plants. Of course, all bacteria also have cell wall, fungi also have a cell wall and plants also have a cell wall. So all these have a cell wall, hence you can take option 2 as the right answer, but still let us cross check the third and fourth options. So third option, because animals is given over there, you can directly cancel it. Bacteria and animals only, animals are there, animals do not have a cell wall, but they possess cell membrane. So it is the outermost membrane, which is the plasma membrane. Cell wall is totally absent in case of animals. So wherever option animals is present, you can directly cancel option 1, 3 and 4. Directly you can click over 2. 2 is the right answer. Next question. Which of the following is not a, question number 29, which of the following is not a character of prokaryote? The option 1, lack of well organized nucleus, presence of 70S ribosome, presence of endoplasmic reticulum and presence of plasma membrane. So, lack of well organized nucleus, which is not a character of prokaryotic cell, they are asking, not a character. It should, so well organized nucleus is not a character of prokaryote, presence of 70S ribosome, presence of endoplasmic reticulum, presence of plasma membrane. So presence of 70S ribosome, it is a character of prokaryote, presence of endoplasmic reticulum. So almost all prokaryotes do not have any membrane bound organelles. So presence of ER is also not a characteristic of prokaryote, and presence of plasma membrane. Of course, prokaryotes have a plasma membrane. So the suspected options are option 1 and option 2. Lack of well organized nucleus and presence of endoplasmic reticulum. So but still among these two we have to pick one right answer. Though organized nucleus is not present, an incipient nucleus is of course present in case of prokaryotes, but endoplasmic reticulum is totally absent in case of prokaryotes. Therefore, we will go with the option 3, presence of endoplasmic reticulum is not a character of prokaryote. Next question. See, this type of complicated questions do appear in your NEET and KCT exams, where you will be put in confusion which is the right answer. You have to think for a while and then answer such questions. Next. Question number 30. A chromosome with one shorter arm and one longer arm is. So, that's speaking about the metacentric chromosome, submetacentric chromosome, the acrocentric, there's a spelling mistake. Fourth is telocentric. There's nothing called as acocentric. Here, metacentric. So, the chromosome has got 4 arms. One shorter arm and one longer arm, they have told. So, the 2 chromatids are joined with the help of a centromere, and if there is a centromere, then which divides the chromosome into one shorter arm and one longer arm, mostly the chromosome is called a submetacentric, because in metacentric the chromosome arms are divided into or of equal size. So there is nothing like short and long, all the arms are of equal size. In submetacentric, one is shorter and one is longer. And even in acrocentric, the one is shorter and one is longer. But one is too short and one is too long there. But here, to avoid the confusion, instead of giving acrocentric, they have given a spelling mistake so that you need not pick up option 3. So you can boldly go with submetacentric, that is option 2 for question number 30. Right answer is option 2. Telocentric, so the centromere is almost at the terminal end of the chromosome, and therefore the right answer is option 2. Next question. 31. Which of the following sequences represents a possible pathway in the production of a secretory protein? So, rough endoplasmic reticulum, secretory vesicle, ribosome, Golgi apparatus. Ribosome, rough endoplasmic reticulum, Golgi apparatus, secretory vesicle. Secretory vesicle, Golgi apparatus, ribosomes, rough endoplasmic reticulum. Rough endoplasmic reticulum, ribosomes, secretory vesicles, Golgi apparatus. So, the right option. Let us look into how a secretory protein is produced. So, the secretory protein is produced by the ribosomes, which are present on the rough endoplasmic reticulum, and finally they are packed up with the Golgi complex, Golgi apparatus. So, rough endoplasmic reticulum will, the ribosomes which are present on the rough endoplasmic reticulum will produce the protein, which are formed into vesicles and finally packed by the Golgi apparatus. So, the option 4 is the right answer for question number 31. Option 4 is the right answer. Next question. Question number 33. 3 of the following statements regarding cell organelles are correct while one is wrong. Which one is wrong? Option 1 tells, statement 1 tells, lysosomes are double membrane vesicles budded off from Golgi apparatus and containing digestive enzymes. Look at the statement, read the statement once again. The first of the half of the statement is absolutely wrong, and the second part of the statement is absolutely right. Lysosomes are double membrane vesicles. They are not double membrane, they are single membrane vesicles, of course. So, the first part of the statement itself is wrong. The second part, of course, budded off from Golgi apparatus is right, and containing digestive enzymes, of course, they contain hydrolytic enzymes in them. That part is right, but it is a single membrane vesicles. So, that is a wrong statement, but still let us check the other statements. The second statement tells endoplasmic reticulum consists of a network of membranous tubules and helps in transport, synthesis and secretion. Of course, it is correct. It helps in intracellular transport and also helps in synthesis of proteins, the RAR, and also helps in secretion. Therefore, that is a right statement. The third statement, leucoplasts are bound by 2 membranes, lack pigment but contain their own DNA and protein synthesizing machinery. Of course, leucoplasts, they are colorless plastids. Of course, plastids, they are made up of double membrane. Of course, leucoplast is a type of plastid, therefore it is double membrane, we agree with it. Lacks a pigment, therefore it is called as leucoplast because it has a colorless pigment, and it is colorless, sorry. Therefore, it lacks a pigment, you can tell, and contain their own DNA and protein synthesizing machinery is absolutely right, because the matrix contains the protein, the enzymes, various enzymes essential for protein synthesis. Spherosomes or single membrane bonds are associated with the synthesis and storage of lipids. So, that also not holds good there. Therefore, the wrong statement is the first one. Spherosomes are of course single membrane bonds and are associated with the synthesis of storage of lipids. Of course, spherosomes, they are not only, they only synthesize, store lipids is absolutely right. Next, we will go to the next question. Question number 34. Which structure is considered as semi-autonomous organelle? So, my answer is mitochondria and chloroplast. Since mitochondria and chloroplast, they can divide on their own by fission and amitosis, they can be called as semi-autonomous organelles, and also they contain DNA and RNA in them, and also contain the various enzymes essential for the cellular activity. Therefore, they are called as semi-autonomous organelles. Next, question number 36. Carrier molecules facilitating transport across cell membranes are. So, cell membrane, we have studied the fluid mosaic model of the cell membrane, where we see two types of proteins, the intrinsic proteins and the extrinsic proteins. The extrinsic proteins are present on the peripheral part of the cell membrane, and the intrinsic proteins are present within the lipid molecules between the two bilipid layer, and they are also called as tunnel proteins since they help in transporting the proteins. So, they are of course proteins, proteinaceous, because there are intrinsic and extrinsic proteins, and the intrinsic proteins which are present between the two bilipid layer, they act as carrier molecules, therefore they are also called as tunnel proteins. That is, the answer is option 1. Next question. Cell membrane possesses, question number 39. Cell membrane possesses lipid, protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the RBCs has approximately, so we have already studied this, the RBC have got lipid, protein and carbohydrates in different proportions. So 40% lipids, 52% proteins are present in case of in RBCs. So 40% lipids and 52% proteins, that is option 3 is the right answer. It is a direct question from the textbook and you can directly point out to the option 3 as the right answer. Next question. Centrosome is an organelle containing two cylindrical structures called cristae, cisternae, centrioles, thylakoids. So, the answer is centrioles. Centrosome has two cylindrical structures which are called as centrioles, and we also have studied the structure of a centriole which resembles the cartwheel structure. Cristae are present in mitochondria and cisternae, they are the part of the Golgi apparatus, and thylakoids, they are present in the plastids. So, the right answer is centrioles, which is related with the cylindrical structures in the centrosome. Next question. 41. Microtubules are absent in mitochondria, flagella, spindle fibers, centriole. So, microtubules are absent in case of mitochondria. So, as we know, mitochondria is the powerhouse of the cell, and there we don't have any cytoskeletal structures. Microtubules are the cytoskeletal structures which are made up of proteins and they are filamentous, which are suspended in the cytoplasm of the cell, and they also form the flagella. They are present in the flagella. The 9+2 arrangement we can see in the axoneme of the flagella, where there are 9 peripheral microtubules and 2 microtubules present at the center, which is called as 9+2 organization in case of flagella. That is the arrangement of microtubules. And microtubules helps in the formation of the spindle fibers. That is, microtubules themselves form the spindle fibers during cell division. And centriole, the cartwheel arrangement, where the peripheral proteins, they are nothing but the microtubules made up of tubulin, the protein tubulin. Next, so the option 1, that is mitochondria do not have microtubules, or microtubules are absent in mitochondria. Next. The membrane bound vesicular structure formed from Golgi apparatus and contain hydrolases. So, the membrane bound vesicular structure formed from Golgi apparatus and contain hydrolases. Hydrolases are the various enzymes which help in the breakdown of the complex molecules. So, they are hence also called as hydrolytic enzymes. And the hydrolytic enzymes are found only in the lysosomes. They are absent in case of ribosomes. Ribosomes are the sites of protein synthesis. Lysosomes, in them they are called contain the hydrolytic enzymes, and of course they are single membrane bound structures which are vesicular, and therefore you can go with option 2 as the right answers. So they contain the various hydrolytic enzymes like the amylases, proteolysis and lipolases. So mesosomes, they are the infoldings of the cell membrane, the prokaryotic cells. Vacuoles, we know they are also of course single membrane vesicular structures, but they do not contain hydrolases. So vacuoles, they contain some waste or excess of water or whatever it is. So hydrolases are present in the lysosomes. So option 2 is the right answer. Next. Question number 44. So sorry. Question number 47. The inclusion bodies present in prokaryotes are all except. So inclusion bodies, phosphate granules, synophysin granules, glycogen granules, all these are inclusion bodies present in the prokaryotic cells. That is, they are the reserve food materials present in the cell in the form of phosphate granules, in the form of synophysin granules and in the form of glycogen. Chromatophores are the pigments which are present in of course, the eukaryotic cells. Chromatophores are not related with the prokaryotes. So, all the three inclusion bodies are the phosphate granules, synophysin granules and glycogen granules. So, except chromatophores, you can take fourth as the right answer for question number 47. Next is question number 51. Keeping in view the fluid mosaic model for the structure of cell membrane, which one of the following statements is correct with respect to the movement of lipids and proteins from one lipid monolayer to the other? So, look at the question. It is based on the fluid mosaic model of the cell membrane, and which of the following statements is correct with respect to the movement of lipids and proteins from one layer to another layer, they are telling, which is also described as the flip-flop movement. So, options 1 and 2, while proteins can flip-flop, lipids cannot. Neither lipids and proteins can flip-flop. Both lipids and proteins can flip-flop, while lipids can rarely flip-flop, proteins cannot. So, here the right option, look into the question. While proteins can flip-flop, lipids cannot. Neither lipids and proteins can flip-flop. Both lipids and proteins can flip-flop, while lipids can rarely flip-flop, proteins cannot. So, we have the fluid mosaic model where proteins act as the tunnels. So, the option 4 is the right answer. There are tunnel proteins on the plasma membrane. Lipids can rarely flip-flop but not the proteins. Proteins can only be transported with the help of the tunnel proteins. So, next question. The study of structure and function of cell and cell organelle is histology, cytology, molecular biology, cell physiology. So, the options are, first option is histology. Histology is not the answer for the above question, because histology deals with the study of various tissues. Histo means tissues. Study of tissues and their arrangement in the organs is called as histology. So, it does not hold good. Cytology, cyto means cell and logy means logos or study. So, the study of structure and function of cell and cell organelles is called as cytology, is absolutely right. So, molecular biology deals with the various biological molecules that are produced in the body. So, molecular biology does not hold good there. And cell physiology or the various mechanisms that takes place in the body, in the cell, that is like cellular respiration or the photosynthesis, final part of the photosynthesis, dark and light reactions that takes place in the mitochondria. All this can be part of cell physiology. So, they are the various mechanisms that takes place in the inside the cell. Therefore, the right answer is option 2. Cytology is the study of structure and function of cell and cell organelles. Next question. 55. Plasma-desmata connection help in cytoplasmic streaming, synchronous mitotic divisions, locomotion of unicellular organisms, movement of substances between cells. So, plasma-desmata or the small openings which are present on the cell wall. So, plasma-desmata connection help in cytoplasmic streaming. It doesn't help in cytoplasmic streaming. Synchronous mitotic division, no. Locomotion of unicellular organisms, no. So, movement of substances between cells. So, this can be the right answer. Movement of substances between the cells is brought about by the plasma-desmata because they are small openings which are present on the cell wall which connect the two adjoining the cells. So, movement from one cell to another cell of the substances can happen through the plasma-desmata, not with cytoplasmic streaming or the synchronous mitotic divisions or the locomotion of unicellular organism. Of course, locomotion of unicellular organisms can be brought about by cytoplasmic streaming. They are related, but the right answer for plasma-desmata connection helping is movement of substances between cells. Next question. Question number 60. Mark the odd one with respect to cytoskeleton. Mark the odd one with respect to cytoskeleton. Maintain shape of the bacterial cell. Network of filamentous proteinaceous structure. Provide mechanical support. Regulate orientation and distribution of cell organelles. So, mark the odd one with respect to the cytoskeleton. Maintain shape of the bacterial cell, network of filamentous proteinaceous structure. Of course, the skeletal structure, cyto means cell, skeleton means the skeleton of the cell. So, they are referring to the various cytoskeletal microtubules like structures present in the cell which will maintain the shape of the bacterial cell. Cytoskeletal structures are not present in the bacterial cell because they are prokaryotes. Network of filamentous proteinaceous structure, of course cytoskeleton are, and they provide mechanical support to the eukaryotic cells is also right. They regulate orientation and distribution of cell organelles is also right. But bacteria is a prokaryote and therefore the odd one is maintain shape of the bacterial cell. It's not the right answer. It will maintain the shape of the eukaryotic cell can hold good there. Therefore, the odd one is option 1, maintain shape of the bacterial cell. Next, question number 62, match the following. So, on column 1 you have the various cell organelles, on the column 2 you have the various functions of them, and the column 1 is telling centriole. So, for centriole what you can match is cell division. So, centrioles they are present only in they are a part of the centrosome and they are present only in the animal cells and they take part in cell division. Therefore, A and 4 you can match. Then lysosomes, they are the suicide bags of the cell because they contain various hydrolytic enzymes and sometimes they break the cell itself, hence they are called as the suicidal bags of the cell. Therefore, B is 1, you can match with B1. And mitochondria, they are the powerhouse of the cell, hence can be called as, it matches with 5, option 5 on the column 2. And ribosomes, they are protein factories which will match with option 3 on the column 2. So, the right option in the answers is option 3 where A matches with 4, B matches with 1, C with 5 and D with 3. So, option 3 is the right answer for question number 62. Next question. 63. Column 1 you have the various different cells and column 2 you have their shapes. So, A is RBC. RBCs are round and biconcave is the right answer. WBCs, they are round and oval. Columnar epithelial cells, they are long and narrow. Nerve cells The option is like amoeboid is there, long and narrow is there, elongated is branched and long. Nerve cells are branched and long, you can go with it. A is trachid. So trachid, they are also long, elongated. Mesophyll cells, they are amoeboid in shape. So the right answer is RBCs round and biconcave, WBCs round and oval, columnar epithelial cells, they are branched, they are elongated, then columnar epithelial cells, they are elongated, nerve cells, they are branched and long, trachid, they are long and narrow, mesophyll cells, they are amoeboid. There's a mismatch. RBCs will go with round and biconcave. WBCs will go with B3, A2, B3, C4, then D6 and E5 and F1. So option 1 will match with the column 1 and column 2. So once again, RBCs, they are round and biconcave. WBCs, they are amoeboid. Columnar epithelial cells, they are long and narrow. Nerve cells, they are branched and long. Trachid, they are elongated. So option 1 matches with the question number 63. Next, question number 65. Single membrane bound organelles are. Single membrane bound organelles are. So membrane bound organelles are of two types, single membrane and double membrane organelles. So you have there lysosomes, peroxisomes, ribosomes and glyoxysomes. So here lysosomes, of course, they are single membranous. Peroxisomes, they are also single membranous. Ribosomes, they are not membranous structures, non-membranous structures, because they have got certain units like 50s and 30s units, and glyoxysomes are also membrane bound structures. So the right option is 1, 2 and 4. All of them, they are single membrane bound organelles. So option 3 is the right answer for this question. Next is question number 66. A few characters of prokaryotes are given below. Identify the number of characters which are not present in eukaryotes. They are not present in eukaryotes, but they are present in prokaryotes. So glycocalyx, of course, it is a character of prokaryotes and not eukaryotes. So plasma membrane, it is present in both prokaryotes and also eukaryotes. Mesosomes, they are present only in prokaryotes. Pili, they are present only in prokaryotes. 70s ribosomes, present in prokaryotes. Plasmids, they are present in eukaryotes and not in prokaryotes. DNA, so they are present in prokaryotes as well as eukaryotes. Cell wall, they are also present in prokaryotes. So 5 of the characters are present in prokaryotes. One is the glycocalyx, the second is the mesosomes, the third is the pili, the fourth is the 70s ribosome, and the fourth is the cell wall. So all these are present. 5 important characters of prokaryotes are seen there. So the answer is option 1. Next, question number 67. Which of the following 4 cell structures is correctly matched with the accompanying description? So they have given the various cell organelles and the description of them. Look into the first statement. Plasma membrane, outer layer of cellulose or chitin. So mitochondria, bacteria-like elements with inner membrane, highly folded and bare electron acceptors. Chloroplasts, bacteria-like elements with outer membrane, highly folded. Then Golgi apparatus, stacks of flattened vesicles. So which of the 4 cell structures is correctly matched and which is the mismatch? Plasma membrane, outer layer of cellulose or chitin they have given. So correctly matched. Plasma membrane is not correctly matched. Yes, mitochondria, not correct, outer layer of cellulose or chitin. It is not the plasma membrane, but it is the cell wall, so it is not matched. Mitochondria, bacteria-like elements with inner membrane highly folded and bare electron acceptors. It's the right statement because the mitochondria are rod-like structures and bacteria are also rod-like structures. They have both inner and outer membranes, and the inner membrane is highly folded to form the cristae. So it is a right statement. Chloroplasts, bacteria-like elements with outer membrane highly folded, and Golgi apparatus, stacks of flattened vesicles. So fourth is also right and the third is also right, whereas the first option, plasma membrane, outer layer of cellulose or chitin, is the mismatched statement. So the three statements 1, 3 and 4 are wrong statements, mismatched. The only right matched statement is option 2. So correctly matched is the option 2. Plasma membrane, outer layer of cellulose or chitin doesn't match because plasma membrane is made up of lipoprotein. Only cell wall is made up of cellulose. Chloroplast, they are not, their outer membrane is not folded, so it is also a wrong statement. Golgi apparatus, stacks of flattened vesicles. Not only vesicles, there are also cisternae and vacuoles. Therefore, it is not complete. Therefore, we will go with mitochondria, which is the correct statement. Next. Question number 70. Common characters found in chloroplast and mitochondria are. Common characters found in chloroplast and mitochondria are both involved in metabolism. We do agree. So, chloroplast is involved in photosynthesis and mitochondria is involved in phosphorylation or ATP synthesis. Both possesses circular DNA. We'll go with that. So, both contain ATP synthetase enzymes. Yes. Both can synthesize proteins. Yes. So, common characters found in chloroplast and mitochondria, all the four A, B, C, D will match. Therefore, option 4 is the right answer for this question. All the four statements will match both for or common characters of both chloroplast and mitochondria. So, option 4 is the right answer. Next. Question number 73. Particles observed by George Pallady are composed of two subunits. Cilia causing the movement of either the cell or the surrounding fluid. The ribosomes on endoplasmic reticulum are bigger than the cytoplasmic ribosomes. So there are three statements given and you have to tell which are correct and which are incorrect. So the first statement, particles observed by George Pallady are composed of two subunits. Of course, it is correct and George Pallady discovered ribosomes. Cilia causing the movement of either the cell or the surrounding fluid. Of course, they are locomotory structures in unicellular organisms like the paramecium. So, that statement is also correct. The ribosomes on endoplasmic reticulum are bigger than the cytoplasmic ribosomes. No, they are of the same size. Therefore, you cannot go with that. So, the first two statements are correct, but the third statement is incorrect. Under the options, you can select the first statement, that is A, B correct and C is incorrect. So, option 1 is the right answer for question number 73. Next question. Question number 78. Which of the following statement is correct? So, ribosomes do not contain DNA. Eukaryotic ATS ribosomes break into 50S and 30S. Plasma desmata are found as intercellular junction between animal cells. Then ribosomes were discovered by Beard and Tatum. So which of the following statement is correct? Ribosomes do not contain DNA. Of course, it is a correct statement. It will not have DNA. Instead, it is divided into two subunits. Eukaryotic 80S ribosomes break into 50S and 30S is wrong. It will break into 60S and 40S. The 70S will divide into 50S and 30S. So 80S will break into 60S and 40S, therefore it is a wrong statement. Plasma desmata are found as intercellular junction between animal cells is wrong because plasma desmata are part of cell wall, therefore they are absent in case of animals. Ribosomes were discovered by Pallade and not Beadle and Tatum. Therefore, only option 1 is correct. So, option 1 is the right answer. Next. Find out the correct option on the basis of the following diagrams. So there is a set of diagram given over there, all related with the structure of the chromosome. In the first, you can see the chromosome structure, which is divided into two chromatids with arms and a central centromere, and at the top you can also the satellite body is seen. In the second, you see the chromatid, and in C you see the chromosome where it is acrocentric type of chromosome, and D it is telocentric. So now look into the options. So option 1 tells that A is satellite, B is secondary constriction, and C is short arm, D is long arm. So, of course, A is satellite, but B is not the secondary constriction. C is the short arm, of course. D is the long arm, of course. But B is not the secondary constriction, but it is centromere. So, in option 2, A is satellite, of course, right. B is centromere, also right. C is short arm. D is long arm. Of course, all of them match properly, but still let us check out third and fourth. Second option seems to be the right answer, but still check out third and fourth. What is wrong in third and fourth? A, secondary constriction. A is secondary constriction, he is telling, but it is satellite, not the secondary constriction. The constriction is the one which is present between the satellite body and the telomere. So, you cannot call that A as the secondary constriction, but it is satellite itself. B is satellite, no, B is centromere. Long arm is C is the short arm, D is the long arm, so it is not the right match. So, nucleolar organizer region, A is a nucleolar organizer region, of course correct. B is not the secondary constriction, but it is the centromere. So, the right option is option 2, which matches with A, B, C, D given in the diagram. Next question. Question number 81. A or granular structure first observed under an electron microscope as dense particles by Dash in the year 1953. Here A and B are. So A are granular structures, means they do not they are not like membranous structures. So A is ribosomes, B is Perner, B option 2 is A lysosomes, B D Dewey, and option 3 A peroxisomes, B Fleming, fourth A ribosomes and B George Pallade. So A are granular structures first observed under electron microscope. It should be ribosomes because lysosomes are vesicle-like structures, peroxisomes also. A is ribosomes, and if A is ribosomes, ribosomes were first found discovered by George Pallade and in the year 1953. So the right option is option 4. A ribosomes and B George Pallade is the right answer. Next question. Correctly arrange the sequence of gram stain in bacteria. So in bacteria, we have gram-positive bacteria and gram-negative bacteria. So, it is based on the staining technique. So, the staining technique is used so that it will absorb the violet color and it will not remain colorless. If they absorb the gram stain and remain colored, then they are called as gram positive. If they lose the color, then they are called as gram negative. So, what are the various steps involved there? Treated with alcohol, excess stain washed with water, stain with crystal violet dye, 0.5% iodine added, observe under microscopic field for violet or colorless bacteria. So, the right option is, so first of all what you have to do is the bacteria culture is first of all treated with stain with crystal violet dye. So, option 3, stain with crystal violet dye. First you are going to do that, then you are going to, excess stain is washed with water. First the bacterial cells will absorb the stain, and then if they are dipped in water, the excess of stain is removed, and with some 0.5% of iodine is added so that it will take the color, and then it is treated with alcohol. So, alcohol is the one which removes the color, and even after treating with alcohol, if the color is there, then you call it as gram positive. If the color is not there, then you will call it, if it is colorless, you call it as gram negative. So, fifth one, then you are going to observe under the microscopic field for violet or colorless bacteria. So, the violet ones are called as gram positive and the colorless bacteria are called as gram negative. So, the right sequence of treating the gram stain is option 4. First, you will go with staining the bacterial culture with crystal violet dye, then you will add, then you will put it in water, stain, wash with water, then you will add 0.5% iodine, then you will treat with alcohol, and if finally all the color, the bacteria becomes colorless, they are identified as gram negative bacteria and it has got absorbed violet color, then it is considered as gram positive bacteria. So, that is because of the thickness of the cell wall or the glycocalyx which takes the color or which does not take the color. So, the one which takes color is called as gram positive, the other is gram negative. Next question. Question number 24, cartwheel structure. Cartwheel structure 84 is seen in basal body of cilia, centriole, both 1 and 2, shaft of cilia and flagella. See, here cartwheel structure is seen only in the centriole, but not in cilia, not in flagella. So, basal body of cilia or the axoneme does not contain the cartwheel structure. The cartwheel structure is present in centriole, and in cilia and flagella you find the 9+2 arrangement of the microtubules. So, 9+2 organization is seen and you do not call it as the cartwheel structure. It is restricted to the centriole where there are 9 microtubules in the peripheral region and there is a central hub, and the hub and the peripheral microtubules are connected with radial spokes in case of centriole, which is called as the cartwheel structure. Next question. Question number 89. Consider the following 4 statements. Golgi apparatus principally perform the function of packaging material and help in middle lamella formation. B. Quasi-fluid nature of lipid bilayer enables lateral movement of protein within the overall bilayer. VL shape of chromosome is seen in anaphase. Then RNA contains 5-methyl uracil. How many statements are correct? Here 3 statements are correct. So, option 1, Golgi apparatus principally perform the function of packaging is right. Quasi-fluid nature of bilipid lipid bilayer enables lateral movement of protein, that is the movement of the intrinsic proteins is also right. VL shape of the chromosome is seen in anaphase, of course that is also right. The chromosomes, that is, which is called as the acrocentric and the metacentric, submetacentric, both of them, they are of V-shaped and L-shaped structures of chromosomes are seen in anaphase, and all the three statements are correct except RNA contains 5-methyl uracil. So that is not a part of the correct statement. So three statements A, B, C are correct. Fourth is a wrong statement. Next. Question number 93. Protoplast is. So what is a protoplast? Another name for protoplasm, an animal cell, a plant without a cell wall, a plant cell. So which is more appropriate? Another name for protoplasm. Protoplasm is the nucleus and the cytoplasm, you call it as the protoplasm. An animal cell, no, in animal cell you don't use this word called as the protoplast. A plant cell without a cell wall. So entire cytoplasm, the nucleus and all the cell organelles together you can call that as the protoplast. A plant cell cannot be called as a protoplast. So the most appropriate statement there which matches for protoplast is a plant cell without a cell wall. So we have discussed more than 45 questions which may help you to tackle your various competitive exams like the CET or the NEET. So we will conclude this video over here. Thank you all.